Hello, everyone. Happy Research Day. My name is Maya Blasingame, and I am a junior biology major on the pre-med track from Baltimore, Maryland. And I am working under the mentorship of Dr. Giannis Tekla. And so today I'm presenting on the unknown world of Pestinella, a whole genome assembly and annotation of the diverse amoebozoa species. So just to start off, I wanted to list the main objectives of the project, given that I was conducting a whole genome assembly of the species, which is Pestinella species is a relatively understudied amoeba species, and this is justifying the importance and necessity of carrying out this research project and doing whole genome assembly. This is really important because it allows the understanding of various genes and proteins that affect Pestinella's role within various environments and ecosystems. And so this objective will allow me to gain insight into the genes and the proteins they encode for. The second objective of this project was to compare the genome and genes found from the whole genome assembly to well-known and related amoeba species and genes that have been found in other species. So I wanted to see, are these genes present in this specific Pestinella species? Does this mean that it's closer or more related to these other amoeba, or does it have diverse genes that are separating it? And this is helping understand a little bit about the evolution. So I wanted to give some general background on amoeba species as a whole. Amoebas play an important role with the environment and serve as regulators of soil populations and other ecosystems. And so their role within the environment is further emphasized because they can act as hosts and reservoirs for bacteria and viruses to survive and transmit diseases, indicating their role in the pathogenic process. Furthermore, in the past, amoeba were thought to be asexual because of their taxonomic category. However, through the discovery of genes and drafts of genomes of other amoebas, this helped understand that some amoeba actually have sexual life cycles. So this is really important because it leads to the question of what genes play a role in this process. What is the diversity of sex genes among different amoeba species? What does this tell us about the diversity of genes of all types of genes among amoeba for a variety of cellular processes? And the second point, as I mentioned, is really important because amoeba are unicellular organisms. And so they were thought that because of this feature, they were asexual. Normally, organisms that have multiple cells or multicellular are going to carry out sexual processes, not unicellular. And so the fact that they have genes indicating their sexual life cycle is significant, and they're carrying out meiosis. So on this slide, I just wanted to show a picture of Pestinella. They're the very tiny dots, not the bigger circles, but the very, very tiny ones. So they're very numerous. So Pestinella are marine amoeba, which means they are found in compost and other marine environments. Furthermore, they do not contain contractile vacuoles, but can survive within saltwater environments. So Vanellidae amoeba are a fan-shaped type and can form cysts, which allow them to have a protective coating and survive within these within these aqueous environments. And so this is important because Pestinella have a lot of these similar traits, even though they're still relatively unstudied. And so this is just a brief video of Pestinella under a microscope. And so they're very, very tiny, as I mentioned, and there's a lot of them. And so this gives you an idea of how tiny amoeba are. But I wanted to further emphasize that although they're tiny, their role within the environment is still so significant. And before I go on to the next slide, I also wanted to talk briefly about the genome size of amoebas in general and highlighting their diversity. So one of the largest genomes belongs to a very, very small organism, an amoeba, and it's called Amoeba dubia. And so the genome is actually 670 billion base pairs. And the genome of Amoeba protis has 290 billion base pairs. Now, these are not, the Amoeba protis is pretty small in comparison to Amoeba dubia genome size. But just to emphasize this point, compared to the human genome only has 2.9 billion base pairs. So even though amoeba are way, way tinier than humans and other organisms, their genome is significantly large. So this highlights not only the diversity because of those differences in the genomes of the amoeba species, but their vast genome size, playing a role in the interactions with their environment and showing how important amoeba are. So, going on to the methods, as I mentioned, the main objective of this project was to carry out whole genome sequencing. And this aims to reconstruct an organism's genome by realigning smaller sequence fragments and reads. As a result, one can then annotate and determine where genes are located, what chromosomes, and what role those genes have just by determining the genes are present. And so, some other analysis that's going to be done or was done included the Busco score, which is a quantitative assessment to determine the completeness of a draft genome based on gene contents and expectations of gene completeness. And the final main analysis and method done was eggnog annotation, which was really important because this is an annotation of new and novel sequences of genes and categorization of sequences based on orthology. And so this allows for the identification of genes present, how many, what types of genes, and orthologs that were present. So I really want to expand and go into detail about the whole genome assembly process. And so I'm going to briefly go through this process just to explain why these steps are important. And this was the majority of my project. As mentioned, whole genome assembly is a reconstruction of an organism's genome by breaking the genome into smaller reads that overlap with each other, and sequencing and assembling the reads in the correct order, which is significant based on these overlapping sections. It can be really thought of as two main parts. The first part is sequencing, which causes a fragmentation of the original but unknown genome to create reads in contexts that have the genes present. The next step is then assembly. And assembly is like putting the puzzle pieces back together in the correct order in order to create a complete picture. This is done because the reads that are in fragments overlap with each other, so the ending of one read will match with the beginning of the next read. And you can see this here in the second box. You see there's blue and red and pink. And so you can see they're overlapping with each other so that you know, okay, this one's going to go first, and then it overlaps with the blue. That must go second. And then you have the assembly part. And so I wanted to break this down more before we get into the results because this slide just shows another overview and flowchart. To start off, you have DNA isolation. So DNA within an amoeba has to be isolated. And so this is the wet lab part of the project. The amoeba are grown, cells are centrifuged, and DNA is amplified to isolate it. The next step is where whole genome assembly comes in by sequencing the DNA using various techniques such as Illumina sequencing, PacBio, or Oxford Nanopore. And actually, a hybrid or a mixture of these techniques are used. And so the sequencing is going to use these different technologies in order to allow for different lengths of reads and for overlapping to occur. For example, Nanopore creates long reads while Illumina is going to create short reads. And after the genome is sequenced, assembly occurs in order to create a full genome once again from the fragments. Assembly is really important because we use hybrid assembly methods from the different technologies in order to account for errors. The next step, as I mentioned, involves polishing, decontamination, and essentially creating a full yet accurate form of the genome. For example, although Nanopore creates long reads, many errors occur, and thus Illumina had to be used to correct for those errors by using short reads in order to match up and make sure everything was polished and decontaminated. The final step, which is really important, is annotation. And so the assessment and annotation of the genome using bioinformatic tools such as Busco and Eggnog Mapper are essential. Annotation specifically allows for a prediction of the genes in the genome, including the percentages, their breakdown, and their categories. So this is one of the most important and vital steps because although we have the genome after assembly, we don't know much about it or able to use much of it unless we have annotated the genome and figured out, okay, what genes are there, what are they used for? So before going into the results, I just wanted to show a picture of some other wet lab processes that I did in this lab. I carried out an ICC staining, which stands for aminocytochemistry staining. So this allows for the visualization and localization of the Passionella species. This was done by staining and fixing the cells to a chamber and then using a confocal microscope to visualize them under various lights such as red and blue light. And so this next slide is just a brief video to show some of the data that was used to analyze the genome. And so this video is cool because it shows all the protein sequences that were present and encoded from the genes and the genes within the genome. And so the length of the proteins vary in size, but this gives a really nice overview of the variety and diversity of the genome. So next we have chronovisualization, and chronovisualization is a tool to understand the taxonomic classification of the genome. It is used in order to understand the evolutionary history and breakdown of the genome and genes present. No hits on this video just represents genes that are unique to the amoeba species or that were not found within the database, but this video further emphasizes the diversity of the genome. Corona is important because it allows you to see if lateral gene transfer has played a role in the presence of genes within Passionella. And lateral gene transfer is the process of transferring genes between two non-mating groups, such as Passionella and bacteria. And so on this next slide, I just wanted to show a bigger screenshot from the corona visualization that I made. And this goes more into detail about lateral gene transfer and the breakdown of genes classified from each organism and the acquirement of genes through lateral gene transfer. So for example, this picture shows that the percentage of genes matched with other organisms, bacteria found in COMP 13%, viruses 0.2%, archaea 0.6%. So this is important because it indicates that a substantial number of genes could have arisen from lateral gene transfer and highlights the importance of coevolution among various species and the acquirement of genes through this process. So moving on more to annotation and assessment. As mentioned earlier, BUSCO scores are used as a means to analyze and determine the completeness of the genome. BUSCO scores were taken before and after decontamination, and I really wanted to preface and highlight that this table was used just as a comparison method, and that pre-decontamination contains any genes and any information about the amoeba cells that could have been infected with bacteria or any other contaminants. Thus, the next slides give the final results from BUSCO. But pre-decontamination, the BUSCO score was 38.8%, and the total length was found to be 58,771,277 nucleotides, indicating this is a fairly large genome, even though the Passionella is very small. The GC content is also an important feature because this percentage actually varies among amoeba species, highlighting their diversity. So as I mentioned, this slide is showing BUSCO scores after decontamination. So the figure on the left is showing the scaffold, figure on the right is showing coding sequences. So scaffold is a structure, proton, or a portion constructed that contains a majority of the structure, including non-coding sequences, like introns, and sequences that do not play a role in proteins that are encoded for. So as a result, this BUSCO score was expected to be similar to the pre-decontamination results and was found to be 37.2%. On the other hand, the coding BUSCO score was expected to be much higher because this is going to contain the genome with the sequences that are coding for proteins, and so this should be complete. And so anything above 75 to 85% indicates, anything above it indicates that the genome is pretty complete. And so this score was found to be 87.5%. And so this is important because this is telling us our genome is complete. I also wanted to highlight the total length, which was found to be 24,968,982 nucleotides, which was less than the slide before just because this was after decontamination. So eggnog. So now we're talking about the genes that are present. Eggnog annotation and mapper was done in order to find the clusters of orthologous genes together and really tell a story of the role of the genes within the genome and how they impact Passionella and allow it to interact with its environment. So eggnog mapper characterized the genes into four main groups. These groups included information storage and processing, cellular processing and signaling, metabolism, and uncharacterized. And so the cellular processing and signaling actually contributed to the highest percentage of genes, which was expected because of the role of these genes in cell signaling, control of processes such as mitosis, cell movement, essentially any process that allows the amoeba to survive and interact with its own environment and other environments. This next slide is really important because it goes back to one of my main objectives, looking at the presence of sex genes within the Passionella species genome. Passionella and other amoebas are unicellular, so they're not thought to carry out meiosis or sex life cycles. However, I looked up gene names that correlate to meiosis and sex life cycle that have been found to be present in other amoebas, such as Cochlepodium minus, Acanthamoeba castanelli, so some really well-known amoeba. And there were actually genes that were present in Passionella species genome and the other amoeba. And so this is really important because the presence of these genes indicates some of the possibilities of Passionella species to carry out sexual life cycles and carry out a lot of other roles within the environment. For example, SPO11 is a really important gene within the sex pathway and the sex gene, and it was present and found to be in the genome. So the final results obtained were the top 5 gene categories, which are shown in this slide. And so these gene categories are essential because they ensure the survival of amoeba in processes such as aerobic respiration. For example, genes were found that encoded for proteins such as succinate dehydrogenase, which is an enzyme within the citric acid cycle, and that's important for getting oxygen and or getting ATP for the cells to have energy. And there was another enzyme that was found that actually encoded for actin and microtubules, which are vital for cellular division and cytokinesis. Moving on to the discussion, a comparison was done earlier to look at the presence of a few important genes and proteins among various species. So SPO11, as I mentioned earlier, is a necessary gene that indicates the process of meiosis and is contributing to the sexual life cycle. And so this is also related to tyrosine kinases, which were present in Pessinella species, indicating its similarity to some well-known amoeba. And so this gives insight into not only the diversity, but also the similarity between these amoeba. As I mentioned, another vital protein is actin, which is necessary for cell mobility and motility, and cellulase. I really wanted to bring up cellulase because cellulase is an enzyme that is used to break down cellulose. And so humans, we actually don't have the ability to break down cellulose. We don't have the enzymes to break down these linkages. And so we can't use cellulose for metabolism. However, Pessinella amoeba species were actually found to have cellulase, indicating their ability to use cellulase or cellulose as metabolism. And this shows just one biological advantage that they have and gives insight into possibly more advantages that they could have that interact with their environment. So to conclude, I wanted to state a little bit more about my discussion, which summarizes this project, which aimed to put together the puzzle pieces of the genome of Pessinella species. Many amoeba have genes that contribute to their sex life cycle and regulated cellular pathways. And specifically, Pessinella species are marine amoebas. And so they were found to have unique genes that not only related to their marine lifestyle, but these life cycles such as sexual life cycle and cellular processes. And so this indicates the importance of the genome and the importance of all these genes playing a role in putting together the picture. Whole genome assembly allows us to identify and tell the story of Pessinella species through the genes it encompasses and uses. It's like, for the first time, figuring out what Pessinella does and breaking into this unknown world of Pessinella species. So some final future steps. There are two main steps that I would like to expand on in the future. And so these are going to include the now sequenced genome. We could use it in order to further study the role of Pessinella in various ecosystems and potentially its interaction with other organisms such as humans or other bacteria. And so an expansion of the comparison of various amoeba species could be conducted in order to create a database to understand how evolutionary history and environmental factors impact the genome and evolution among various amoeba species. And so thank you so much. These are my references, and I wanted to say a huge thank you so much to my mentor, Dr. Giannis Tekla, Han Tran, our lab tech, Ms. Brandy Taylor, the research day chairs and judges, and all the sponsors for taking the time to really listen to my presentation and helping put it together for me. This wouldn't have been possible without any of you guys. Thank you.